Okay, so in this first video, we're going to look at blood vessels. So we're looking at the three major blood vessels, or technically five major blood vessels. Then we're going to take a little look at tissue fluids, different fluids in the body. So first up, the three types, or the three major types of blood vessels. So arteries, which carry the blood away from the heart, veins, carry it back to, and capillaries. They effectively are the site of exchange. And so a couple of details about each one now. In terms of the kind of nomenclature and the language that we might use, then tunica interna, tunica media, tunica externa might not be the type of language that you might see in the books or necessarily in the revision guides. And so you may see it referred to other things, but one thing that is pretty much uniform is the lumen, which is effectively the hole in the middle of the blood vessel. And then in terms of arteries and veins, they have a tunica interna, so an inside bit, a tunica media, a middle bit, and a tunica externa, an outer layer. With capillaries, they effectively only have the inner layer, and this being a tunica interna. So, if we take a look at the structure of each of them, so the tunica interna being the endothelial layer, this single layer of cells in here, and effectively a capillary is just that, an endothelial layer. One thing that you'll notice in terms of the lumen, in terms of an artery and a vein, the lumen's much wider in a vein. This reduces friction effects, and so the blood can flow easier. And one other thing that you'll notice is the valve present in the vein, and so that's required to stop the backflow of blood, particularly from blood returning from the lower limbs, from the legs, back up to the heart. The middle layer here, so the tunica media, is basically two sections. It contains a smooth muscle layer, and it also contains elastic fibres. And the thing that you should notice with an artery is the elastic layer fibre is much, much thicker because it has to withstand higher pressures, has to be able to recoil as well to be able to maintain the blood pressure, whereas the valves, that layer is much thinner. The muscle layer is much thicker again in the artery because it's likely that an artery has to be able to either constrict or dilate, and so that muscle layer enables the lumen size to be altered, and therefore you can redirect blood down certain arteries or arterioles compared with veins, which have a much smaller layer. The outer layer, which is referred to as the tunica externa, that tends to have things like collagen fibres in as well, and that can basically is a strengthen and aid. And the other thing to mention about collagen is that's also found in the tunica media layer, particularly the elastic layer fibre kind of section. So as a bit of a run through, tunica interna we can think of as just the endothelial layer, and that's probably the type of thing that you'll see in the revision guides and in the books. The tunica media, smooth muscle layer, and also the elastic layer as well. So the elastic layer containing things like collagen. And the tunica externa, again, this is the outer layer, mostly comprising of things like collagen fibres for strength. So in terms of looking at some of the sections here, so what we can see here is a capillary bed. And so obviously the capillaries are much, much smaller than the other blood vessels. But one of the things that I mentioned at the start with the five blood vessels is arterioles and venules. So an arteriole is effectively a small artery. So what you would have is the blood flowing down an artery, and then that's going to branch out into smaller blood vessels, arterioles. It will reach then a capillary bed, and then it will those capillary beds will come together to form a venule, and then those venules will collect together to form a larger vein, and then get back to the heart. So in terms of looking at the blood vessels in terms of blood pressure, then it should be no surprise that the further you get away from the heart, the lower the pressure. So if we take the run through here of the major blood vessels, so the aorta is the closest to the heart and therefore has the highest pressure. Then the arteries, the next major arteries are the next biggest blood vessels. So again, we can see that the pressure is beginning to drop here. Arterioles much smaller, and then at capillaries has a reasonably small blood pressure. Venules smaller still, veins smaller still, and the vena cava the lowest of them all, because again, it's the furthest away from the heart. One thing to notice about this graph as well is the difference between, you can see this variation that you get here, and this is the difference between the diastolic pressure, so the pressure of the blood going through the aorta and the arteries under relaxation when the heart is relaxed, and then under contraction, as obviously you would expect when the heart contracts, the pressure is going to go up. And so that's what this variation is here. In terms of venous return, so venous return is just a name meaning blood returning to the heart through the veins. Now, the biggest problem for this is the fact that the veins have to carry blood back from the lower limbs back up to the heart, and so have to work against gravity. And one of the things that can force it back through is obviously the movement of the blood being basically pushed around. But another thing that can really help this is the contraction of the large skeletal muscles in the lower legs and the upper legs. So the blood vessel, the vein, is found pretty much nestled between kind of large muscle groups, and when these muscles contract, they squeeze the blood vessel, and by squeezing the blood vessel, they're going to force the blood in an upwards direction. Now, what would happen if the blood gets forced in the upper direction? This valve, which is designed to let the blood flow this way, will open, let the blood through. But this valve, if any of the blood goes downwards, snaps shut. So it prevents the blood going to the bottom of the legs, in effect. But this movement, or these contractions of these muscles, really helps return the blood back to the heart. And without the valves as well, none of this would be possible. So, a couple of other definitions that might be used here. Anytime you see the word pulmonary, that effectively means a circulation going to the lungs, and systemic effectively means going to the rest of the body. So let's have a look at the different types of fluids that are produced. So of the 3 types of fluids that we're talking about here, we're looking at blood, we're looking at tissue fluid and lymph fluid. So we'll take each one in turn. So obviously you'd be expected to make a list of the types of things that you would find in the blood. So effectively the blood is a mobile carrying device. It takes stuff around the body and it delivers it to certain areas, and then it takes other waste products and delivers those to the areas where they need to get to. So in this regard, the types of things that you would find in the blood, you've got red blood cells, white blood cells, you've got blood plasma, you've got water, you've got all of the nutrients, so any of the complex carbs, simplistic carbs, sugars, you've got proteins, amino acids, triglycerides, pretty much everything you could think of is that cells require is delivered to them in the blood. Now, the question is, if it's found within the blood, what you need to get is you need to get it inside the cells. So the question is, how can we make this as efficient as possible? And how that's done is the formation of this tissue fluid. And what tissue fluid is a fluid that will bathe the cells found in a particular organ or in a particular tissue or location. So, for instance, if you looked at the muscle cells that are respiring at the minute in your body, then the tissue fluid is formed around those, and it's basically a fluid that bathes those cells in a nutrient-rich broth, if you like. So, effectively, all of the things that are required by the cells can be pushed out into the tissue fluid and therefore can be supplied to the cells. So it is effectively a liquid that surrounds the cells and allows a transport of the types of nutrients and the types of substances that are required by the cell. Now, in terms of how it's produced, it's an interaction between these two forces, and in this case hydrostatic pressure and osmosis. So let's have a look at what these terms actually mean. Hydrostatic pressure. So this is the pressure of the blood, or in this case any liquid. So it's effectively the pressure of any liquid that could be exerted. So in this case, we were to look at this blood vessel, then there's a hydrostatic pressure because the fluid moving through this blood vessel is going to exert a force, and that is effectively the hydrostatic pressure. And then the fluid on the outside of the vessel pushing the other way is that fluid's hydrostatic pressure. So we can see on this diagram that the pressure pushing out is greater than the pressure basically pushing the other way. And so we could say our net hydrostatic pressure in this case will be 3.5 kilopascals going out. Now, one of the things that you have to be very, very clear on is that where this is happening is where the tissue fluid is formed is at the arterial end at the capillary beds. And because they're porous, they allow certain substances out. Then the substances that are allowed out of the blood vessel are particularly small. So if you think back to what the constituents of the blood are, the things that are allowed out here will be small enough to squeeze across some of the gaps in the porous membrane of the capillary. Anything too big won't be able to get out. So for example, very large proteins would be too big to squeeze out here. Amino acids can fit through. Red blood cells, too big to squeeze out, whereas something like glucose is small enough. Things like, obviously, carbon dioxide and oxygen are able to be squeezed out of these blood vessels, whereas anything large will not. So, the other type of force at play is the osmotic pressure. And so the osmotic pressure is, is effectively down to the net loss of water from a particular area. So in this case, the osmotic pressure on the arterial end acts in this direction. It's going back inside. And so what we have is this. We have our hydrostatic pressure, and the net hydrostatic pressure is basically out 3.5. And then at this end, the osmotic pressure is forcing stuff back in. So we can say at the arterial end of the capillary, because the hydrostatic pressure is much higher, it's going to force stuff out of the blood. So the stuff in the lumen gets forced out here. Now, if we compared that to the venular end of the capillary bed, you can see here that the hydrostatic pressure has obviously dropped massively, but the osmotic pressure has remained pretty much consistent. And so if we look at the values here, then relatively speaking, you've got one going out here and then two going in here. So the overall pressure is going to force stuff back into the venular end. So anything that's collected, that's been produced by the respiring cells, can then be placed back into the capillary bed here, and then can be emptied into the vein and could be carried back round. Now, if we look at this as a whole, the blood is forcing some of those materials out here. So things like oxygen, things like water, things like amino acids, triglycerides can be forced out of the blood and forms the tissue fluid that bathes these cells. At this end, because the hydrostatic pressure is dropped from inside the blood vessel and the osmotic pressure is greater, here it's going to force things back in. So anything produced by the cell can then go back into the venular end. However, if you look at this, there is obviously a difference between these two values overall. And so basically some things that are not reabsorbed back into the venular end will go on to form this fluid, which is called the lymph fluid. And this is effectively everything else that's left over that hasn't been reabsorbed at the venular end. And so this would include some amounts of oxygen, some of the waste products that you might, or trace amounts of the waste products that you may well produce here as well. This lymphatic fluid will then be kind of recycled round, and the name, the lymph vessel, should imply that the other thing that's going to be moving around in these types of vessels will be lymphocytes. And so what will happen is these lymph vessels basically run side by side to some of the other types of blood vessels, so some of the larger arteries and veins, and then the lymph vessel will empty back into a large vein and therefore go back into the blood system. So if you look at this, the lymphatic system, then you can see that these vessels pretty much would run side by side to some of the larger blood vessels like arteries and veins, and they actually empty into the veins so that stuff can be recycled back round. Now if in this case you have an issue with some of that lymphatic fluid, then what would happen is you would end up with issues like this. So with a high degree of lymphatic fluid that will not drain back into some of the blood vessels, then you can get this swelling, which is referred to as an edema. And effectively this is down to the fact that some of these lymphatic vessels have been damaged. You're unable then to recycle. Go back to this. So because some of these lymph vessels have been damaged, they're unable then to feed back into some of the major blood vessels. You get this pooling of the lymphatic fluid, which results in that swelling, that edema. And there's one thing to just kind of review what would be going on here. So In terms of what you would find and where, erythrocytes is a posh name for red blood cells. So you obviously would find that in the blood. You wouldn't find it in tissue fluid and lymph because it's too big to get out. Lymphocytes, again, dependent on whether you have an infection or not, you definitely find it in the lymphatic fluid, and you might find it in small amounts in the blood and small amounts in the fluid if you had an infection, for instance. Plasma proteins, again, they would be found in the blood. You probably wouldn't find you'd find very few of them in the tissue fluid and the lymph, and again, because they're large. Oxygen, now this depends on which end that you're looking at. If you're looking at the arterial end, then you would expect to find high levels in the blood. At the venular end, you might expect to find smaller amounts, but still some. Tissue fluid, you might expect to find a very small amount of it in the tissue fluid at the arterial and the venular end, and you might find a small amount of it in the lymph. Carbon dioxide, likewise, at the arterial end, you're likely to find smaller amounts, and at the venular end, you're likely to find more because obviously your cells are producing it. In the tissue fluid, you're likely to find trace amounts because some of it's going to be not recycled back into the venule, and then therefore that's why you find a small amount of it in the lymphatic system as well. Okay, hopefully that's been of help.